Good morning, and thanks for giving us a chance to present our project, Discovery of Neutral Allosteric SHP2 Inhibitors through Ensemble-Based Consensus Molecular Docking, Endpoint, and Absolute Binding Free Energy Calculations. In this project, we were trying to find novel enzyme inhibitors for the SHP2 protein that is known to be overexpressed in several malignancies. Inhibitors targeting the allosteric site of these proteins are currently at the forefront of the drug discovery efforts for small molecules against this protein. However, current inhibitors are still in the clinical trials, and the majority of them are known to interact with the hERG ion channels, meaning that they will be most probably cardiotoxic. Our aim is to find novel set of commercial molecules that can be further optimized to clinical candidates that don't have this hERG inhibition limitation. To achieve this goal, we have performed a computational screening followed by in vitro activity assays. For the screening, we developed a three-stage screening protocol. One, pharmacophoric screening for the known chemical libraries using the pharmacophoric features extracted from known SHP2 ligands. Two, docking-only screening for commercial libraries without taking advantage of the known SHP2 ligands. Three, combining the output of one and two, and performing additional round of screening with two docking software, four scoring functions, and two different protocols to calculate the binding affinities. We ended up by getting 26 small molecules that were purchased and tested here at U of A. For the screening assays, two different types of assays were performed. One, a binding assay to confirm the engagement of the small molecules to the target, and two, activity assays to confirm the ability of the small molecules to achieve the desired biological outcomes. For the binding assay, we performed the thermal shift assay, where a small molecule binder will increase the melting temperature of the target protein, as we can see here in figure 2. For the activity assays, we have performed two different types of assays. One, an apoptotic assay to measure the cell-killing ability of the small molecules against cancer cell lines. We can see here that indeed some of the molecules achieve a moderate activity in this assay compared to the control. For the second assay, we measured the level of IL-2 release from CD4 cells, which is a known biological outcome of SHP2 inhibition. And we can see here that some of the molecules indeed resulted in moderate IL-2 release. At the end, we were interested to see if the observed activities of the selected active molecules correlate with their binding affinities and the stabilities of their molecular dynamic simulations trajectories. And this was indeed the case. The molecules can form all interactions and even more, formed with the known ligands without requiring the presence of the quaternary nitrogens, which is a known structural alert for hERG inhibition. So in conclusions, our research identified three active molecules, 1, 2, 3, and binding kinetic analysis demonstrated that these molecules have nanomolar range of affinities against SHP2. These molecules are free from the tertiary amine group associated with many of the current allosteric SHP2 inhibitors, which usually form quaternary ammonium group at physiological pH, a known structural alert for hERG inhibition. The identified compounds are chemically stable, have been pre-filtered for the presence of pan-assay interference compounds, and are amenable for future rounds of medicinal chemistry optimizations. And thank you.